And the way the bacteria transfer these factors among each other, that varies. Sometimes the bacteria just get together and actually transfer the factors. But they can also acquire these factors through viruses that may move between bacteria carrying these pieces of genetic material from one to another, or they can even just take the pieces of the genetic material from the environment. So there are different ways that bacteria can actually share these resistance factors, and then we end up with a big problem of a vast number of bacteria that we no longer can treat with the antibiotics that we have. So this is why we have sort of this campaign now to be a little bit more aware of the problem of antibiotic resistance, and know that we should not take antibiotics unless we have a confirmation that indeed we have a bacterial infection. So just in case is not something that we should do, or we shouldn't take antibiotics to treat viruses because viruses are not killed by bacteria. And also, if you do need antibiotics, it's important that you complete the whole treatment to prevent those mildly resistant bacteria that may be in your body, prevent those from surviving and then coming to recolonize the populations. And also help to prevent transmission by washing your hands frequently. And this is especially important in hospital settings where there are a number of people infected by all sorts of different kinds of bacteria. So thinking about the principles of evolution by natural selection can help us understand how these antibiotic resistance evolve. The same principles apply when we are thinking about treating viral diseases, like, say, the HIV virus with ACT and a variety of other drugs that we use for them. The viruses also evolve resistance to these drugs, and we have to come up with the strategies to minimize that resistance from evolving and figuring out a way of getting rid of them more definitely from the beginning. So the other was an example of evolution that is happening within our bodies, but involving cells that are not part of ourselves. Now, it turns out that evolution by natural selection also happens among cells in our bodies. And in one of the cases, this involved the development of the immune response, which is a good thing for us. But it also happens to happen in the case of the development of cancer, which is actually not a good thing for us. So let's think about a little bit about the immune response. And so here is a puzzle. We are large-bodied organisms that live for a long time, like 100 years or something like that. And we are, in a sense, in competition with these pathogens that are infecting us. And these pathogens can evolve very fast to become adapted to our bodies and do damage to us. And the reason why they can evolve very fast is because they have very short lifespans. They have huge population sizes and high mutation rates. Because evolution depends on the amount of genetic variability that is present in a population. When you have these huge population sizes and these high mutation rates, there is already a lot of variability to start with that natural selection can use as a raw material for evolution. And so as a result of these, viruses and bacteria can evolve as much in a day as we can in 100 years. So how is it that we can exist at all when we are being affected by these really fast evolving organisms? And the answer to that is because we have been able to match our pathogens one-to-one in all these accounts. And that is done by the cells of our immune system. And so the cells in our immune system are actually evolving by a process of evolution by natural selection during the immune response. And the cells in our system they have short lifespans, they have huge population sizes, and they also have very high mutation rates, and I'm going to show you how that works. So with these properties of the cells of our immune system, we actually have leveled the field in the competition with our pathogens. And what the cells in our immune system are evolving is a very specific response to particular pathogens. So that when we are exposed to a given disease at any point in time, our immune system learns to recognize the disease, and next time we are infected by the same pathogen, it is not going to make us sick anymore. And it's the same principle that applies when we are vaccinated against some particular pathogen. So how does this work? So we have two different, and this is going to be a highly simplified view of a very complex system, which is the immune system. But basically, we have two types of cells in our immune system, those called T cells, which recognize infected and damaged cells in our bodies and eliminate them. So cancer cells, for instance, would be subject to destruction by these T cells. And then we have the B cells that produce antibodies, which are basically these proteins that attach themselves to pathogens to tag them for destruction so that the immune system can recognize them as pathogens that need to be killed. And what's really interesting about how this works is that these antibodies are tailor-made for specific pathogens. So there is going to be antibodies developing for the flu virus, for instance, that are going to recognize the specific features of the flu virus and actually match it like a lock and a key. And that way, it can very specifically recognize a particular disease and eliminate it right away the second time we encounter the same pathogen. And also, this immune response can also allow us to keep up with the pathogens when they are evolving within our bodies. So how is this accomplished? And this is actually a really fascinating problem and a really fascinating solution, because we are faced with thousands, hundreds of thousands, or even millions of possible pathogens, pathogens that exist now and pathogens that are going to exist in the future. The antibodies are coded in our genes. They are proteins that are actually being encoded in our genes, what the sequence of these amino acids that make these proteins are going to be. But we don't have genomes that are large enough to account for every possible pathogen that we have. So the way this is done is actually by producing, through a process of genetic recombination and mutation, a population of B cells in our bodies that are highly variable, and then choosing among that highly variable population of cells those that are a better fit against particular pathogens. And I'm going to show you how this happens. So our B cells, that are the ones that are going to produce these antibodies, they have in their genome the sequence of genes that are going to encode the variable region of the antibody that is going to match the pathogen. And there are 3 different segments that get assembled in this variable region. And in the genome, there are different possible alternatives for each of these 3 segments. So there's a few dozen of these, a few hundred of those, a few hundred of those. And during the process of cell division, they undergo recombination in this region of the genome to produce different combinations of these possible different kinds of segments. So that now we have different cells that have different combinations of the different segments. And I think the calculation is they can make about 28,000 different combinations. But on top of that, they undergo additional mutations in these genes in the genome that add to that variability so that then they can actually have almost every possible combination of possibilities here so that they can find a good match to a particular pathogen. So once we have produced a very highly variable population of B cells, and that is represented here, we are just showing 3 different types, but you can imagine here there are going to be thousands or hundreds of thousands of varieties there. Then these B cells are producing their antibodies, and those antibodies, so these are these little things sticking out of the cell here, are then being tested against these features of the pathogens that the cells are going to recognize. And those that end up being a good match are the cells that are going to survive and reproduce. So a little bit like what happened in the case of the bird that was picking up the green versus the brown beetles. In this case, the immune system is picking up those cells that are a good match to our pathogens to be the cells that go on to survive and reproduce, and the other cells that are not a good match to the pathogens, or cells that may perhaps be matching some of our features in our cells, those are eliminated from the system. And at the end, we just end up with a population of B cells in response of a particular infection that are now a good match to a particular pathogen. And these cells are going to produce very highly specific antibodies that are going to help us fight that infection. But at the same time, we are going to maintain in our bodies these so-called memory cells that are going to have the memory of that specific pathogen. So that next time we get infected by the same pathogen, these memory cells are immediately going to recognize this, and then they are going to expand again and start producing antibodies that are going to eliminate infection, probably without us even noticing that this has already happened. So this, again, is a process of evolution by natural selection happening within our own bodies. And we are lucky that we have this system. Actually, we probably couldn't exist in the way we exist if it wasn't because of this adaptive immune system having evolved sometime in our evolutionary past.